Bismillahirrahmanirrahim, and welcome back to this next video. And this is yet another video in the series of videos on the bioinformatics. In my previous video, I've told you about the genetic code table, and I've told you how the genetic code table it works and which kind of the information it gives you. I've told you that three nucleotides they combine with each other to give you a single codon, and a codon it codes for an amino acids. So this particular table is going to give you 64 codons, 61 of which are coding for the amino acids, and 3 like the UAA, the UAG, and the UGA, these are the stop codon. That means these are signal to stop the process of the protein synthesis further. I've told you that the genetic code table it is actually made up of these three letters, like the first letter, the second letter, and the third letter, and when you combine them, that is going to give you codons, and each codons they are specifying a single kind of the amino acid. Now in this particular video, I want to focus on how you can use this genetic code table. Now, from a given starting point in your DNA sequence, this is the most important thing which I'll be focusing in my next video, that the starting point in the DNA sequence, that is very important, and that is going to give you information about the primary sequence of a protein. So if you start from one point, you are going to get one kind of the protein. If you are going to start the same DNA sequence from another point, you are going to get a very different kind of the protein, which I'll focus in my next video. But for this particular video, if you are going to start from a given sequence, from a given point in your DNA sequence, you are going to start reading the sequence 3 nucleotide at a time. And these 3 nucleotide, which is of course one triplet, this is technically known as the codon. What you do is, if you have identified a codon, then you have to consult the genetic code table, and the genetic code table is going to give you the information that this particular codon is going to code for this particular amino acids. For example, if this is the messenger RNA sequence, and this is the, say for example, this is thymine. In my previous video, I've told you that most of the softwares that you use in the bioinformatics, they usually do not most of the time differentiate between the thymine and the uracil. But if you are talking about the messenger RNA sequence, of course this T is going to mean the uracil. If you are dealing with a DNA sequence, this particular T is going to refer to the thymine. So don't worry about that. So if this is the DNA or the messenger RNA sequence, what you have to do is, the first thing you have to do is to split them into triplets. Like this is ATG. So this is going to be one triplet, and this triplet is technically known as the codon. So this is one codon, this one is another codon, the another codon, and so on. So you have split your messenger RNA sequence into the successive triplet codon. Now you have to consult the genetic code table, and you have to see that this ATG or this AUG for which amino acid it codes. So if you are then consulting the genetic table, this particular sequence of the nucleotide or these codon, they are going to give you the corresponding amino acid sequence or the primary sequence of that particular protein. For example, if you look at this one, this is ATG. So this ATG is going to code for the amino acid methionine. And as you can see over here in the genetic code table, of course, you are going to see the AUG. Here it is ATG. So that is one and the same thing, because instead of the thymine in the messenger RNA sequence, you are going to get the uracil. So this ATG, it is going for the methionine. This GAA, this is the glutamic acid. If it comes at the GAG, so this GAG also code for the glutamic acid, because one of the property of the genetic code table is it is redundant. By that I mean that more than one codon can code for an amino acid, and this is just one of the example that the glutamic acid is encoded by 2 codons, like the GAA and the GAG. Then you have got the GTA. The GTA is going to go for the valine, the phenylalanine, and so on. So if you are then going to split it into the successive codons, so each codon is going to give you the information, or that is going to give you a specific amino acid. And in the end, if you can see, this is the stop codon. In the genetic code table, I've told you that there are 3 stop codons, or if there is a stop codon in the sequence of the messenger RNA, this will be the point where the translation machinery is going to stop the synthesis of the proteins. So if your DNA sequence is correctly listed in the 5' to 3' orientation, you will generate the protein sequence in the conventional N to C terminus as well. I'll continue this discussion in the next video. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.